Welcome to the PharmaSource podcast, recorded on stage at CDMO Live Europe 2026 in Rotterdam. This episode is sponsored by PharmaBlock, a fully integrated CRDMO providing research, development and manufacturing services from early discovery to commercial production.
I'm Jeff Mason. I am the Vice President of Sales at Samsung Biologics. You're probably also wondering why the head of sales is up here talking about tech transfer. It's a great question. I almost think of the TikTok meme. You're probably wondering how I got here. So as Ben mentioned, I have about 30 years experience in the CDMO space. Started out in manufacturing for about 22 of those years. So I've kind of seen a lot of tech transfers, probably on the order of hundreds of tech transfers over the years. I've seen the good, the bad, and the ugly for sure. Today we're just going to talk a little bit about who Samsung is. I know many of you are familiar with us, but we do get a lot of questions quite often about what we do. Obviously everybody knows the traditional Samsung brand, but we're going to talk a little bit about that, then we're going to get into the tech transfer case studies, and then talk a little bit about the future approach that we're going to be taking at Samsung. Okay, so Samsung, you probably know by the electronics industry. You probably have some appliances at home for sure. We're involved in electronics, semiconductors, engineering and construction, finance, and of course mobile. Quick humorous story. Even yesterday, I had a lovely woman, come up to the booth. She looked through our pamphlets and things. She looks up at me. She takes her phone out of her purse and goes, "Is this you?" "Yeah, that's us." I thought she was going to actually ask me to fix her phone. Thank God she didn't. So the thing about us, though, is in 2011, we decided to get into healthcare and biotech. And since then, we've really been able to accomplish a massive amount. We've got about 145 global clients, 17 out of the top 20 biopharma companies. We've got 440 plus regulatory approvals, and adding to that each and every month, really. 55 successful inspections from the FDA. We have a 99% success rate on the manufacturing floor for DS. And we employ now about 5,800 employees, both in Songdo and now at our newly acquired site in Rockville, Maryland. A little bit of the timeline here. We were founded again in 2011. We started DS manufacturing in 2013, and closely behind that we started DP manufacturing to offer the full end-to-end solution. We added development services, so it was a little bit backwards. Sometimes you start with small and you work your way up. But we started with large capacity and sort of worked our way backwards to development services. But we do offer the full line all the way from cell line construction through commercialization. mRNA services we got involved with because of the pandemic, and we do have those available as well. And then last year we started our ADC business, so we have a separate facility for that, and we're completely capable of providing GMP batches at large scale for ADCs as well as development. We added the organoid services, so in the early research and development stage, just last year as well. And then most recently, just at the end of March, at the beginning of April, we acquired a Rockville, Maryland site from GSK. So that's a fully operational site, employs about 500 people, and we will continue to put in new processes there and use it as our first footprint on the U.S. soil. This is just showing you the complete capacity of Samsung. We are very large. We have the most capacity, essentially, in the world. Most of this is all, again, located in Songdo in South Korea. We have 5 different plants for manufacturing within 2 bioparks. There's space for a third biopark that we can add to, as well as 3 more plants to the existing biopark 2. We have scales ranging anywhere from 1K all the way through 5K, 10K, 15K, and now our newest acquisition, we also have 20K reactors available. Along with that, we have DP lines that we can provide both liquid and lyo product to you. We will be adding a pre-filled syringe line next year as well. All right, so getting into the tech transfers. So when you think about tech transfer, and I realize some in this room maybe are not at that stage yet, but I do encourage you to start thinking about it, but also thinking about a second source. Like, "Oh, Jeff, I just want to get one source. I don't need 2 yet." But it's really important to consider at least up front, because it really can save you a lot of money, time, and heartache down the road. You will have a robust supply chain at the end, potentially in 2 different scales, so it allows you to be much more flexible with your forecasting, etc. But it doesn't come without complexity. It certainly introduces a little bit more in a time that you're trying to get your product to market. However, I think the payoff is worth it. But you do need to consider such things as transferring across 2 different sites at different scales. That can introduce certainly a large amount of variability, whether it's in differences in mixing, whether it's in mass transfer, or other process dynamics within your program. What we're seeing, though, is a trend for a lot of our customers have an increased demand to do a dual-site tech transfer because of accelerated timelines, because of hedging risk for their supply chain for commercial. And this doesn't really allow a lot of time in order to process optimize, to align the critical quality attributes and characterization across the sites. And that can all lead to increasing the risk of an out-of-spec result as you transfer in to the receiving site. So any delays or inconsistencies can obviously add major headaches to yourself. You can risk batch failure, which then can delay even batch release, but more importantly, getting to market. So our approach at Samsung is a structured dual tech transfer. This is a risk-based approach. It's a parallel strategy that enables rapid and consistent implementation. So the reason I showed you all the capacity and everything is I wanted to give you an idea of how we're set up. So when you look at those sites and those plants, they're in close proximity of each other. They are also built in a very similar fashion, almost mirror images of themselves, whether it's a 10K scale, 5K scale, 15K scale. They're very similar, and they're built on the same type of quality platforms, et cetera. So you do have to consider such things as supply chain management when bringing in 2 different sites at the same time. You're going to have to look at the major process gaps and equipment gaps. Again, the way we are designed, this eliminates a lot of that risk. You want to look at streamlining your supplier qualification for your key raw materials. This can often be a huge roadblock to any tech transfer, so it's something that you really want to take a close look at and make sure that you can supply both sites simultaneously. Of course, CapEx is always a consideration. Are there any different changes you need to make in engineering at any of either the receiving or donor site during tech transfer? And if you do, then you need to make sure that those are aligned. Analytical method, this is where you can really save time, the way we are designed, the way our biocampuses are designed. We have shared laboratories. So really you only have to do 1 tech transfer, even though you're going to end up with 2 different nodes. Saves a ton of time, as you'll see in the timelines coming up. So you want to make sure that you got the proper tech transfer expertise in your sending unit. This is extremely important. By being within the same company, that helps a lot. A lot of times it's the same team, so basically they end up just talking to themselves. But you want to be able to deliver a comprehensive documentation package at the end of the day so that the receiving site can take in the process in a proper manner. And then you want to transfer critical materials and process knowledge in a very efficient way, structured communication and coordination between the 2 sites, and again, our design allows that to be quite simple. So what we call it is excellence. This is our dual sourcing strategy. It's really designed for speed, standardization, and reliability. Again, it comes down to our plants being carbon copies, cookie cutter, if you will, from one to the other. Whether it's a 10K or 15K, it's all run on the same platform, has the same quality systems, same raw material management systems, et cetera. It's really a data-driven, uniformed platform with 430 regulatory approvals across all of our different scales. And then the regulatory approval is fueled by geopolitical proximity. So what does this all add up to? This adds up to a parallel site process validation, which is, for you, the important part is that it's less time, less cost, and regulatory becomes much simpler. And you don't need to put as much resource towards 2 different separate tech transfers. This gives you an idea of the timeline of a standard tech transfer. Typically, our standard tech transfer, you can define it different ways on when you stop tech transfer, but typically it's at the beginning of the first GMP run or just after the first GMP run. So typically at Samsung, we're running about a 6-month tech transfer from tech transfer strategy discussion all the way through to the first GMP run. When you're doing dual site tech transfer, because of, again, the way we're set up, this allows us to start that tech transfer while we're still progressing through the initial one. What we're able to do in that case is really omit a couple of these steps. You're already going to have your tech transfer strategy discussion up front, so that's not needed for the second tech transfer. The sending and receiving units are closely linked right away at the very beginning, so they're well aware of all the different gaps that we may have bringing the process into Samsung, so they're able to resolve those quite easily. It's all about coming down to one process control strategy, which I'll talk about a little bit in a minute. But the huge piece here is the number 3, and that's really the no need in the second site to have an analytical method transfer, because again, you're using a shared centralized laboratory to do all of your QC testing. It's a huge advantage in terms of time. So we're able to do that in 90 days. And we proved this a couple years back in 2022 and 23, and this is just giving you an idea of that timeline. And we started in November of 2022 with the first plant. This was a 15K plant, and we transferred into a 10K plant and later on that year. So we did this simultaneously. We proved that we could do it in 90 days, and again, this was measured between the first batch out of freeze and the first batch out of freeze in the second plant. What this gives you ultimately, as you can see, is 2 fully qualified nodes within 1 year's time. So you're ready to go through filing, you're ready to get your product launch, you will have 2 nodes ready to go. You could have 2 separate scales, which allows you, again, forecasting flexibility, especially at launch, but beyond that, throughout the product life cycle. Just to give you an idea, okay, Jeff, so do they compare, though? Does a 15 and a 10K compare? So here's just some simple data showing you the viability between the 2 different plants, the 2 different scales. As you can see, they're pretty much spot on in terms of viability and growth curve. The second one does show a slight difference, but we'll call that process variability. I went back and forth with my MSAT guys on that one. And it's not statistically significant. It's a very low end number we're looking at here. So essentially, these are the same at the end of the day. I mentioned it before, but basically our scheme of multi-site tech transfer is simultaneous multi-site transfer enables us to leverage one single process control. So because, again, those plants are all designed the same way, they run in the same manner, they have the same quality platforms, they have the same QC testing, etc., this allows for very streamlined validation and quick movement through the timeline. We can actually apply then this one process control strategy across not only just the 1 and 2 plants, but we can apply it to all 5 of our plants if we wanted to through all the different scales. So what we're doing a little bit beyond this, we are actually doing this as we speak in Songdo. Validation is happening right now. They're going through some clinical runs, and they're doing a dual transfer from one 15K to another with a process that we have there. And the expectation is that we will, again, be able to achieve another 90-day tech transfer into the secondary site. So what this does, using the one process control strategy, is, of course, it reduces process validation efforts for multiple studies. It streamlines your regulatory submissions in a lot of ways, and it maintains product quality and operational consistency between the 2 sites, both receiving and donating. Okay, just to give you an idea, we're actually doing 5 different projects at the same time this year, doing this dual site tech transfer. We're doing a lot of these because there's a need for it, of course, but also so that we can really accumulate our process knowledge. Really, the goal is to improve on 90 days and to make it even shorter, and to do it truly in parallel without any flaws or issues. So that is the end goal. So why we drive to this strategy is for obvious reasons. It's about reducing batch requirements, which end up being a cost savings for you, ends up being a time saving for you, gets you to market faster with a faster launch. You can cut down on your validation and streamline it. And then it also ensures that you have consistency between the 2 sites, which is not an easy feat when you're talking about 2 different plants, 2 different companies, etc. So that, I believe, is the last slide. So I thank you for your time. I know that was a lot of information. And again, I'd be kidding myself if I thought I could cover tech transfer in 20 minutes, but I tried. So thank you very much.